Hello everyone. Biotechnology is employing and harnessing biology and generating different techniques out of it that can be employed for the benefit of human beings and other animals. Now, when we are dealing with biotechnology, we discuss a lot of advanced tools and techniques that we have been discussing in our session. An important technique in this regard is DNA microarray. Now, you might have heard of gene chips or DNA chips, which is also known, which are also the different names for DNA microarray, and it involves basically a slide with attached high density array of immobilized DNA oligomers or cDNAs. So, what we are basically doing it is that for all these techniques, the one property of DNA that is utilized is the complementarity of DNA. So, we have been discussing DNA fingerprinting techniques, and even in this DNA microarray, the basic property of DNA that is utilized is that the two strands are complementary to each other. So, if we know the sequence of one strand, we can ideally generate another strand that can actually go and identify and detect or whatever we need to do for the complementary strand of itself. So, the same thing, another development is the DNA microarray. So, this is attached with the high density array of DNA oligomers or the cDNAs as well. Now, when we say cDNAs, we will see that this technique is mostly employed for knowing the differential genetic expression in different cells. When we say cDNA, it means we are dealing with the complementary DNA. So, this complementary DNA is generated from the mRNA, if you can recall. mRNA is a reflection of the expression of a particular gene. So, when we want to do the expression analysis, when we want to study the transcriptome, that is the mRNA that is generated in a cell, we convert it by using the reverse transcriptase to cDNAs, and then cDNAs can be also utilized in the DNA microarray technique. Now, this represent usually on the DNA microarray, this immobilized DNA oligomers, they represent an entire genome of species under the study, or sometimes it involves a set of genes. So, it depends upon what do we want to study, but most of the times they represent the whole set or entire genome of the species under the study that is involved. The technique rely on the hybridization properties of nucleic acids to monitor the DNA or RNA abundance on a genomic scale in different types of cells. Now, the important property is the hybridization property of nucleic acids, the complementarity. It can monitor the DNA molecules as well as the RNA abundance, because a DNA-RNA hybrid can also be formed. Now, each DNA is attached to the solid support, a glass, plastic, nylon or a silicon wafer, which is most frequently used in DNA chips, and they serve as a probe for binding to its unique complementary DNA, that is the target DNA. Now, this terminology can be a little confusing, so I will emphasize on it and explain it a little bit again, because generally what we do is the entire genome when we digest, then that is termed as the target DNA, and when what we use to identify our gene of interest, that is called as the probe DNA. But in case of DNA microarray, it is in the opposite case, just in the terminology thing, because the DNA that is basically attached to the slide is termed as a probe, and the complementary DNA that we use is called as the target DNA. So, just the terminology is a little different that is utilized here. Some people use it otherwise also, but this is the general thing which has been used in different, since the inception of DNA fingerprinting, that the probe is basically what is on the DNA chip, and the target DNA is our gene of interest or the unique complementary DNA that we use to hybridize to the chip is basically there. So, this is how basically the DNA hybridization happens. Now, what is the mechanism involved? This is a broad overview of the mechanism. The first thing is the preparation of the chip. So, as you can see, the chip can be, as I said, a glass slide or a plastic slide or a nylon membrane or basically a small silicon wafer, and then on that the different DNA fragments are basically bound. We will elaborate on this a little bit more. The second step is the hybridization thing. So now this is the probe which is bound here. Then we use the target DNA. As you can see here, the target DNA is used from two different sources. They are labeled differentially. You can use different fluorescent labels here, and that is used to hybridize the chip. And then this chip is scanned, because wherever this complementary DNA is going to bind, it is going to produce a signal. So we can scan these chips, and after scanning you get the images where these fluorescent bands are giving rise to the different kind of signals. And then these images can be subjected to the analysis part, where we normalize and where we analyze the data by different methods. So this is an overview of DNA microarray, how this happens. Now we will look at one by one things in a detailed manner. So the first aspect is the building the chip. So how does the chip is built? If the chip contains the entire genome of an organism, then we cut the DNA into smaller fragments, or we use the entire genome, and for example in this case we are using the complementary DNA. So we express the genome in the yeast, and then whatever mRNA is produced we convert it into cDNA, and we use these cDNA clones and we put them on different wells on a well plate, for example, and then pins are used. So these kind of pin needles are used to place these individual cDNA molecule which are in the wells and make a pattern on the chip. This is a glass slide which is used. So on glass slide, the 600 set of cDNA clones are basically loaded onto here. They are pinned onto here, they are marked onto here by using specific pins. Certain things are used in duplication, etc., but a pattern is created. So this is how the chip, this is a glass slide how it looks like. The gene chips are even thinner, they are just like wafers, the silicon wafers, and on that surface they contain many number of, so even if entire genome, they will have thousands of oligomers, DNA oligomers, and the entire thing is going to represent either the entire genome or the entire transcriptome, and based upon that these kind of patterns can be generated. So this is how the chips, so different types of chips are available. Now there are different companies that produce these chips. So the labeling or the marking or all these different kinds can be done simultaneously, and the technique that they use for making the chip, putting the DNA on the chip, what kind of oligomers are used, what kind of patterning is used here, these may vary from one chip to another. But the basic thing is that, and again you can also get chips related to, for example, you want to study only a different pathway. So you can basically have the DNA oligomers only of the regions that is involved in a particular pathway or something like that. But they have to be in large numbers, or they have to be, in a duplicate or triplicate they are produced here, so that the authentication is there. But usually the microchips are used for the entire transcriptome, for the gene expression analysis, or for the genome analysis where the entire genome is loaded onto the chips. So this is how a DNA microchip or DNA chip or gene chip looks like. This is again showing the same thing in a different pattern. So this is how the chip looks like. These are the locus, loci of the different DNA fragments. These are different places. On each fragment there is a multiple series of fragments that are contained, and that fragment is going to contain a different type of a sequence. So these are so it can be, on one, when we say the entire genome, then different oligomers of the genome are contained and they are pinned onto here, and they will contain oligomers containing different sequences. So different sequences are in different wells. Of course, they can be duplicate and triplicate things, but then the entire chip represents the entire genome, or they contain the DNA fragments from the entire genome, or as I said, cDNA clones from the entire transcriptome. So this is how the chip looks like. This is how the location of the position of the DNA oligomers look like. These are the DNA oligomers at that locations, and these are the DNA sequences that is shown here. So this is how the chip is built. The second step is to hybridize the chip to the target DNA. Now, in the previous examples, the cDNA clone was shown, but another method I have just included this again. This is building the chip only, but I have just included it here because there can be methods where we can amplify different regions of the genome and then use these amplified products and place it on the pins and pin it on the DNA chip. So these kind of things are also used. So we have annotated means we know which gene is from where and whatever. So we use the annotated genome, we amplify the different PCR products, and then we can use these PCR products and load it onto the chip as well. So we can make this kind of chips also, apart from cDNA clones or entire DNA fragments done by restriction digestion, etc. And as you can see, what happens with the hybridization? For example, we are taking from two different cells. So two different cells, we produce and prepare the single stranded fragments and we label it with different dyes and then we hybridize it onto the chip. Now, whichever oligomer is complementary to this particular DNA or probe or the target DNA, that probe is going to bind to it. Similarly for this case. So these are two different cells, but they will have two different sets of fragments. So even the genome of the two will differ. If it is a transcriptome, the mRNA will differ. For example, we can use the cells of a diseased or a non-diseased state, a normal or a cancerous state, or two different situations that if we are giving a stimulus, we are not giving a stimulus. So when we give a stimulus, transcriptome will differ. We do not give a stimulus, transcriptome will differ. In the diseased, undiseased state, cancerous, non-cancerous, everywhere the transcriptome will differ, and therefore the complementarity of the fragment, the set of fragments will differ, and therefore they will bind to different probes on the chip and give rise to different kind of fragments. This is elaborated by using the different kind of dyes. So here again we are using two different cell lines. In these cells we are isolating the DNA and we are hybridizing it with different fluorescent label. For example, here it is used a red label and a green label, and these red labeled and green labeled fragments are then hybridized onto the chip where each well is containing a set of fragments. Now this is going to bind to the set of fragments. Again, this is showing that how this occurs. So these are the DNA fragments which were present in the well, and these are the fragments that we are using as a target. This is our target and these are the probes that are bound, and we are using these targets to go. Now they will go and bind only to their complementary sequences which are present here. So the earlier example which was showing that the next mechanism is the scanning and the detection. So what happens here, that these are the wells. Now in these wells you can see the different kind of detection patterns. For example, the red is going to bind at these places and equally. So the red fragments, the red fluorescence labeled fragments is not able to bound to any of the fragments present in these two loci or two positions, but they are getting bind to these two positions. Now if we look at these things, then in the green scientific fragments this is binding to these two and it is not binding to this one, and this is binding very lightly to this one as well. So when we superimpose these two images, so this is the image of the chip when only the red labeled fragment is bound. This is the image of the chip when only the green labeled fragments were bound. When we superimpose this chip, we get superimpose these images, we get this kind of a pattern where this shows that this is particularly the fragments are only hybridizing to one, coming from one cell, and similarly this is coming from another cell only because this is red and green, and this particular probe that we have used is bound to both of them. Now the probe we are putting on it, we know either the sequence of it or we know the regions, that what is that. But when it hybridizes, we say that this is the particular thing which hybridizes with both the cell. So this particular cDNA or the complementary DNA or genome is present in both the cells. However, this is only expressed in this cell line and this is only expressed in this cell. And this particular probe is neither expressed if it is the cDNA clone and neither present if it is a genome that we have used in either of the two cells that we have used to compare. So we scan, these images are produced, these images are superimposed, and then signals are used to detect whether the target sequence is present at that particular loci or not. We can then use the fragments at that loci and use it and sequence it, or we can analyze it by functional analysis to understand more about them. But this is how the scanning and detection occurs. So this is again explaining the same thing, that certain where the red fluorescent dye will bind, at certain places no fluorescence will be bound, and at the certain places there will be hybridization of the probes containing a different fluorescence, and therefore when you detect it you get different images. So this is an image, for example, and then it will be further analyzed. So as you can see that at certain positions it is showing red color, at certain positions it is showing green color, and at certain positions it is showing white, yellow fluorescence, and at certain positions it is black, there is no fluorescence at all. So because the DNA chip is known, if it is not even known, then we can identify and take out the fragments from whatever position we know, because the pinning is done by known either the known set of DNA, or even if the sequence is known, we understand that this set of fragments is present only in the cell that was labeled with fluorescence, red fluorescence. This is only with green one. This is expressed in both the cell lines, and some of them are not produced at all. So these are the kind of images. This is again a zoomed out image. You can see very clearly the red fluorescence, the green fluorescence, the yellow fluorescence, and no fluorescence. So yellow fluorescence basically means that the cDNA, if we are using cDNA as the probe, then the cDNA, the probe which is present on this particular loci is expressed in both the cell lines. The red ones in only one cell line, and in green one in only the other cell line. So these two cells can be cancerous, non-cancerous, or under certain condition that we are giving a stimuli. So in the normal cell it is not produced, but when there is a stress, when there is a given stimuli, then that is going to be expressed. So this is the differential expression that we can see if it is the if it is a cDNA analysis, and this is the differential presence or absence of a particular genome sequences or different sequences if it is the genome that we have put on the microchip. So this is how the image comes in. Then after the image is produced, after image analysis occur, because there has to be a normalization, there has to be a clustering that needs to occur, and all these things, because we are not dealing with 1, 2, 3 or 4 genes that we observe. There will be some 6,000 genes that are present on this chip. So for 6,000 genes to analyze, we need and we have softwares that basically first of all normalize the signal so that any background signaling that is coming is basically optimized and removed. So there are equations by which we normalize the signal intensity that is coming, and after normalizing the data is analyzed, the inferences are made, and these data, the image is produced as TIFF images, and this TIFF images can be opened in any of the image analyzer softwares which undergo all these steps and store the information related to either the differential gene expression, or as I said, presence or absence of something, or involved in a particular pathway if we are dealing and analyzing with a particular pathway. So all those different kinds of things are done. So this is the entire process and mechanism of DNA microarray that is produced. Now on the screen is a DNA arrayer. As you can see, these are the array things, and on the right hand side you can see that the pins are used to make the chips. So this is how the pinning, so each pin will contain a drop or a molecule of, a drop of DNA containing different oligomers, and then they are basically placed onto the chip. So this chip, as I said, can be the slide, nylon, plastic, or a silicon wafer. This DNA that is produced here then cross-links to the whatever the amount the chip is. So this cross-linking can be done by different chemical methods, because the chip can be covered by a particular chemical structure that cross-links with whether the DNA is also that thing, so that it binds very tightly and a particular position only. We often use the duplicate and triplicate samples so as to later on say with authentication and use it for normalization as well, the signal intensity, taking average and etc. So this is how the microchips are produced, and this is a DNA microarray. This is a hybridization chamber. So once the DNA microchip has been prepared, and they already come prepared, they are commercially available. You can have the entire genome chips, or you can have the DNA chips also for the entire transcriptome of different species, and these chips you can use your target DNA, your gene of interest to hybridize here. And for the hybridization, these are the hybridization chambers. So you can see that these are, for example, the chip area, these are the glass slides, and maintaining different temperatures, different optimum conditions such as appropriate humidity, temperature, as well as lowering the melting temperature. The different target DNA can bind onto the different oligomers that are present here. So obviously the oligomers are single-stranded oligomers that have been immobilized on this DNA microarray, and then the probe, the target is used to bind to these probes and different buffer conditions and hybridization conditions. You can see that the basic principle of hybridization involved in all the technique is the same, just like we discussed in a general DNA fingerprinting, or in fact southern hybridization. That technique, if we start with the basic technique, it is the same thing. The DNA now just has been, we are not transferring the DNA by conventional method, but the DNA has been transferred by using specified advanced techniques. It has been immobilized, and then the target sequence is used to hybridize to it, and after hybridization, these are the microarray scanners. So the chips are put in here, just like this, the chips are put in here, and these chips, they will scan, and they will record the signal intensity, because the target sequence that we use are labeled by different fluorescent nucleotides, fluorescent labels. They when you scan these microchips, when you scan these microarray chips, then they identify or they detect those signals, and they produce the images that I just showed, and they store that images as TIFF images. Then those images can then be further analyzed in different softwares to give values for example, the differential expression, or absence or presence of different regions, etc., or the presence of different sequence modifications that are there. So this is how the microarray scanner is there, and it works. Now talking about the applications, this DNA microarray technique can be applied to measure the transcript abundance. This transcript abundance is basically termed as the expression profiling, and the cDNA arrays are used to it. This is the most common used of DNA microchips, and in these DNA chips or DNA microarrays, we have the entire transcriptome of a particular cell on the chip. So this transcriptome is basically the mRNA converted into cDNA. So these cDNA transcripts are basically loaded onto the DNA microarrays, and on the microarrays, and this can be done for two different situations, two different cells, two different experiments, and these DNA chips are available. Or in fact, this DNA microchip or the the probe is basically common. For example, we are studying yeast. So if we are studying yeast, we will use the yeast transcriptome DNA chip. and we will prepare the sample by using two different sets of experiments and prepare the target DNA and label them in the differential situation. So, the microarray will contain the cDNA from the yeast or from any genome that we want to study or any cell that we want to study, a normal cell. And then in, for example, the cancerous state we will see, and we will hybridize, prepare the target and hybridize, and we just now, as we have discussed, we detect and check that. So, this is how basically the expression analysis is done by using microarrays. Then the microarrays can be used for genotyping. So, genotyping is again an important thing, that if we want to identify a unique pattern. So, genotyping is basically when we identify the presence or absence of certain important genes that tells us that what kind of genome it is. So, this genotyping can again be done. We can use the entire genome of a particular, we can use the organism's genome which we want to study, and we can hybridize it with the microarrays of different genomes, and we can use the genome, and we can then identify the genotype, and we can do the genotyping of that individuals. We can estimate the DNA copy number by doing what is called as the comparative genome hybridization. So, if we took the genome of two different individuals and we take the DNA microarrays or chips of these two different individuals, and we hybridize one gene of interest to these two, which is present in these two individuals, but at the number of places it is going to bind the different, and if it is represented, each oligomer is represented only once, then we can actually estimate the DNA copy number in the two different individuals. So, by comparing doing a comparative genome hybridization, we can estimate the DNA copy number. We can also determine the identity by descent, which is termed as the mismatch sequencing. So, this is genome mismatch sequencing which is done. So, if we can actually do the lineage, we can identify the identity of an individual and we can see that how much mismatching it is showing. So, we can actually determine to some extent the phylogeny based upon the DNA microarrays, that if the ancestor and the progeny, how much amount of mismatching is there or how much similarity it is showing, we can do the determine the identity by descent. We can measure the mRNA decay rates as well. So, again mRNA decay is basically differential mRNA expression. So, if the mRNA is expressed but decaying, its shelf life depending upon that, we can also note that in two different microarrays, that in one, if the mRNA is not present, then it is in the decaying faster, but if mRNA is present in one place, then it is basically present there. We can also identify protein binding sites. So, where on DNA, where the proteins are bound. So, we can use the DNA chips and we can use the proteins to actually see the labeled proteins, and we can hybridize it to the chips, and we can identify where these proteins are binding, and therefore we can identify that. We can also determine the subcellular localization of gene products. So, we can use the proteins, or we can use the proteins and identify that where they are present in different cells. So, we can use the entire cellular, or even we can use the protein chips, we can use the DNA chips for identifying the using the proteins, that where it goes and binds and how they are produced, and we can determine the subcellular localization also, that whether they are localized in a particular organelle or in another organelle depending upon the kind of proteins or the from where we have isolated them. So, there are multiple applications for the DNA microarray as well, and it is a very, very strong and robust technique. Of course, this is the basic principle and basic application that we have discussed. It has been modified and more advanced, and modification has been done to make it more robust, more friendly, and more sensitive for using in the experiments that we want to do. So, that is all about the DNA microarrays. Thank you.